Thank you for joining us. I'm Merrilyn Datta. The notion of precision health has been with us for decades, but are we finally at the point where we can be precise about its meaning? For young Sophia and her family, it means after years of struggling for a diagnosis for her disease, through whole genome sequencing, they were able to identify a variant and diagnose her with beta propeller protein-associated neurodegeneration. Precision medicine means genomic technology can have a critical impact on a patient's health. And the promise of precision health relies not just on technology, but on a combination of infrastructure, partnerships, and the collaborative spirit. Our panelists today represent a spectrum of voices across the healthcare landscape. We've invited them to shed some light on the topic. Kaja Wasik is the co-founder and chief scientific officer at Variant Bio, which is a Seattle-based startup developing therapies to improve global health by studying the genes of individuals and populations who are outliers for medically relevant traits. She's a firm believer that human genetics has the power to transform drug development. Dr. Jill Hagencord is a board-certified pathologist with subspecialties in molecular genetic pathology and pathology oncology informatics. She's the chief medical officer of Optum Genomics. Prior to joining Optum, Jill was the chief medical officer of several genomics and precision health startups, such as Invitae, 23andMe, and Color. Dr. Paldeep Atwal is a widely recognized expert in medical genetics. He's a board-certified, fellowship-trained, clinical and medical biochemical geneticist. He holds a medical degree from the University of Glasgow and trained in hospital internal medicine with the Royal College of Physicians at Glasgow Royal Infirmary. He completed a genetics fellowship at Stanford University and a medical biochemical genetics fellowship at Baylor College of Medicine. It's a pleasure having you with us. So I think maybe one of the things to do is to level set about what we mean by precision health. There's a lot of terminology out there. So maybe I'll start by asking you guys, how do you define precision health? Jill, why don't you start?
That is an excellent question. I wish I had a simple, straightforward answer for that. But it's the kind of term that you have to define every time you use it, because if you're saying it on a podium and your audience has 100 people in it, there's 100 different ideas of what precision health means.
Kaja, how do you define precision health? How do you think about it from your standpoint?
I think what it is for me, it's really an individually curated approach to diagnostics, to interpreting diagnostic results, and to designing therapeutic interventions for a patient. So it really has to, in my opinion, take into account a person's genetic makeup, their disease history, and a number of environmental factors. That's it.
Great. Paldeep, how about you? How do you define precision health?
Yeah. Well, I have to say I like that individually curated statement. I might use that in the future. But for me, it's about looking at genomics as part of the kind of multi-omic medicine. So it's genomics, metabolomics, proteomics, etc., and combining them to give a deep and rich understanding of what's going on in the patient, and using that information for their personal benefit, individualized to them, not kind of generalized to what would be beneficial on a population level, but for their individual set of needs.
Kaja, can you tell us a little bit about the research at Variant Bio and how you guys are thinking about precision healthcare?
Yeah, so in simple terms, we are using genomics to identify novel drug targets and novel diagnostic targets. And more specifically, we are really working with groups around the world that have a genetic architecture that's conducive to rare variants rising to higher frequency. So that means groups that went through a genetic bottleneck, that live in isolation due to cultural or due to geographical reasons, or maybe groups that have higher levels of consanguinity, because what that causes is those genetic variants that are rare everywhere else can rise to a pretty high frequency, and some of those can be associated with a phenotype that can have consequences to health in those groups. We really like this setup for projects in scenarios like that, because you end up working with a group that has a much more homogeneous genetic background and that is very often exposed to the same environment. So similar diets and other environmental factors, which really, from like a perspective of genomics discovery, makes the signal to noise ratio lower. And this homogeneous genetic background and those variants being at a high frequency also allow us to work with much smaller sample size numbers. So we really have the statistical power to make the discoveries that require a couple thousand individuals or less, rather than hundreds of thousands of individuals that are typical for GWAS in multi-ethnic cohorts. What I'm hoping is that by designing studies this way, that really focusing on well-stratified populations, like well-genetically stratified populations, we can make discoveries that are going to be transformative, because what we're trying to find is single genetic variants with strong effects that can be used for developing therapeutics, because from a therapeutics development perspective, if you want to develop a drug, you want to target a product of a single gene and you want the effect to be robust.
Great. How interesting. Thank you. You know, Paldeep, you're kind of right there on the ground with where precision health is today and being practiced today. Tell me a little bit about your perspective and kind of what you're seeing in precision health.
I can already tell you 6 months ago, I was doing something different to what I'm doing today. The biggest change is obviously in the diagnostics and the power of the diagnostics, the ability to get things like exome sequences, genome sequences, and other kind of omics type tests, even metabolomic profiles, continues to improve, continues to be easier. They continue to be more useful in terms of their clinical utility. And we're starting to find answers for patients that they've been searching years, decades sometimes for answers, per the introduction you had earlier in this in this session. And then even for the exciting part is even we're starting to see patients back now. Let's say they had a negative exome a year or two ago. On the reanalysis or on a follow-up genome, we actually find an answer. That even though they had an exome or something in the past, that gene wasn't discovered, or that disease association wasn't made at that point. So it's really exciting, and I think I'm only seeing it increase, and I'm only seeing it the pace of it increase. So it's very exciting to be part of.
Jill, how do you think precision health is being viewed kind of across, the health spectrum, across many different hospitals and companies? You've been CMO at a lot of different places. So what do you think is the broad perspective of kind of the healthcare industry on precision health?
If you're talking a health system, it always depends on like what percentage of their patients are still in a fee-for-service model versus a value-based model. Because in my mind, the precision health or population precision health kind of model, inherent in that is that you're practicing value-based medicine and your objectives are improving on the quadruple aim. And so that's really, I think, what the health industry is moving in that direction, but it's hard because you're not already there. But for the sake of this conversation, let's pretend that we're successfully moving away from that fee-for-service model. And then I think, the health systems really want to see your traditional health care, ROIs, but they also are starting to pay attention to some different kind of value drivers, things like patient satisfaction, engagement, the ability to recruit new patients in to their health system away from a competing health system, and the ability to retain those patients. And so we're seeing, some of the health systems out there making, a lot of impact if they've implemented some kind of genomics program. If you go to the DC market, you'll see that Innova kind of bombards the airwaves with almost like every single commercial is Innova talking about their precision health and how they do genomics in their health system. And that kind of puts pressure on the neighboring health systems to also figure out, how do I implement genomics? How can I say that I'm doing precision medicine?
What do you think the vision of where we're going is? If you were to look 5 years out, where do you think the biggest changes will be in how we think about precision healthcare, or where the biggest impact will be?
Yeah, so I can think of a couple of different things with that. So number one, I think the way that we do it has to change and has to become more proactive rather than reactive. Many times it's reactive. Someone had cancer at a young age. Someone has a child has some developmental issues. There's some unusual finding in someone suggestive of a rare disease or syndrome, and then they're referred into care. I look at the example of newborn screening as one of the best examples of precision medicine, in that you're able to diagnose a condition before it really manifests, and then if you treat the condition properly, the negative health consequences don't actually come often. So you're really getting ahead of it. The other big thing, I think, looking at other omics together, it's kind of like different parts of a jigsaw. So there's a baseline genomic sequence, but also there's the metabolomics, the kind of real world biochemistry that's happening in the patient. There's the epigenetic profile, there's the transcriptome, etc. And you kind of have to have it all together to really understand and get that deep, rich picture of what's happening in the patient, and then use that to guide their health and management. A lot more. So yeah, ultimately a lot more preventative, a lot more getting in front of potential medical consequences than reacting.
Do you think we're in a good position to bring all the omics together when we think about, where artificial intelligence is going and even just our data infrastructure? Because clearly the ability to bring in the different omics and, do correlations with a clinical outcome is going to be really, really important. So what do you think we need to do to make sure that's in place?
Yeah, I'm not sure if it's a good position, but certainly a better position we've been in the past. You need adequate data sets, and you need adequate sharing of those data sets with appropriate safeguard measures when it comes to things like privacy and control of access. You also need to understand some deep phenotypic data on those data sets to really make sense of what you're seeing. And then using technologies like machine learning and AI to try and find connections that take so much computational power that it's going to be prohibitive to have it done manually.
Yeah, Kaja, you mentioned that in the research that Variant's doing, you're looking for really well stratified populations to kind of get around needing to have super large populations. Is there an area, either from a therapeutic or a particular type of population, that you see that's going to be especially helpful as an approach? Or is there an area that you think over the next 5 years you see coming to fruition because it's super stratified?
Yeah, I think there's a couple of things around that. So what we really focus on is populations that are outliers for medically relevant traits. So we don't just go to a place that's, this is an island, that will have an isolated population. Let's just sequence everybody there. We try to focus on phenotypes of relevance, and then we have our different areas of priority. So we are focused on common disorders. We're focused on complex disease, but disease that really the areas of disease where there's not a ton of different therapeutic targets available. So areas of unmet medical need, like kidney disease, different types of neurodegenerative or neuromuscular disorders, where we're focusing on autoimmune disorders, where we think like having those really well stratified populations is going to be extremely helpful in identifying novel targets. But that also kind of, the reason we're trying to do that is because the data sets that we are working with currently are a little bit messy, and they are really not perfect. And that that is both in terms of the genetic data sets and the phenotypic data sets that are even in kind of like a worse condition. Because when it comes to genetics, there's the data that we have access to, that we can use as researchers to make discoveries, they're very fragmented. They're built on an outdated technology, genotyping arrays, which kind of offer a very narrow overview of the genome. They don't allow for things like novel genetic variant discovery, for instance, and all those databases that are like the the ones that we have access to as researchers are also really consisting of individuals of European ancestry. So they're just not really representative to global genetic diversity, which leads to things like ascertainment bias in our studies, and which leads to things like misdiagnosis because a genetic variant that was associated with X in somebody with European ancestry doesn't really translate to the same thing for somebody with a very different ancestry. So being able to gather better, deeper, more like overarching phenotypic information is of extreme use to being able to really understand what are the functional consequences of that specific variant. So I agree with very deeply that things like metabolomics, things like proteomics, because most of the data that we have access to really rely on a couple measurements. Obviously, you can't over phenotype an individual. If somebody is a research study participant, they don't want to spend hours participating in your research. There's a limited amount of blood you can draw from one individual. There's a limited amount of blood biomarkers you can measure in that individual, and there's kind of limited amount of non-invasive tests you want them to be submitted to before they want to leave and go their own way. So really focusing on phenotypic areas or data gathering areas that can give you a lot of information with relatively low participant burden, like metabolomics, is definitely key. In growing those data sets is going to be of extreme use to make genetic discoveries and help stratify patients for different disease areas.
If you guys, as you think about the challenge both of getting the phenotypic data and the other omics data in a standard way, because obviously some of the challenge is that we don't always standardize, how we talk about phenotypes. But if you think about that, and also the challenge of, kind of merging data sets and being able to query them, if you could wave a magic wand, or you could say, there's just one thing that would make the biggest difference in us moving forward in being able to bring those genotypes and phenotypes together in a productive way, what would be the thing that you would say, we must come together and do this? What would it be for you, Jill?
This is actually one of the major initiatives that my team at Optum and UHG is focused on, is building off our existing assets at Optum, our data assets, which at this point are heavily enriched in EHR, claims data, prescription data, as well as, through our population health program and other programs. We've got social determinants of health, environmental information, some behavioral information. And if you look at, you go to a population health book and you look at chapter 1, there's going to be a graph or a table that kind of shows you what are the determinants of health. And depending on which study they cite, the numbers are going to be right around this. It's about 10% of what determines our health is in our medical records and claims data. About 30% of it is genetics, about 30% of it is social determinants of environment, and about 30% is daily behavioral choices. And Optum has been collecting all of these other types of data assets and turning them into useful products for our healthcare industry partners. So once we start adding in the genomics data to that, and there's two routes to that, right? There's the raw genomics data, but then there's always just the test results. This person had a oncotype DX, what was the score? And then we have the outcomes data to say, that patient was supposed to get X treatment. Did that actually happen? If so, why or why not? We are going to be in a position where we can make not only process improvements to the delivery models, but also, better understanding of the real world utility and real world health economics of different precision medicine interventions.
Paldeep, what about you? If there were one thing you could do in either how we collect data or or the infrastructure that we use to house the data that could move us forward, what do you think that would be?
I think the answer lies in both of the things you just said. So that how we collect it and what the infrastructure is that is being collected on, and how that infrastructure can actually aid in the collating and understanding of it. So if you look right now, we have these kind of population databases, as Kaja and others have already pointed out, that there's limitations. There's often just genotypic data and other limitations, poorly phenotyped. And And then there's security concerns as well. So what you really need is a database that has a extremely large data set that anyone can add to in a structured fashion that can then be interrogated by appropriate individuals. And it's not just genomic, but it houses other omics data. And then there's some format that you can actually take that data, use it in applications such as looking at biomarkers or patterns that predict cardiovascular disease outcomes. But that it's all kind of separated right now, and it's actually you can't do that. So something like that would really help accelerate some discovery.
You know, Kaja mentioned the fact that a lot of our databases mainly contain genomes and genomic data of, kind of Western European ancestry. I think also as we think about data and infrastructure, we have to think about how we pull in larger diversity of data. Do you think that'll have a large impact? How important do you think that is in the mix of, really getting the data and infrastructure correct?
It's hugely important. And it's sometimes falsely assumed it's important. For example, having more African ancestry or having more Asian ancestry will be useful for those populations. That's actually part of the truth, but it's actually just as important, arguably more important for other ancestries to have that information in there, because it helps you understand the diversity of human variation. Most of the diversity is actually in African populations. So we have a lot of learning that will benefit everyone to look at the wider diversity of human variation.
So each of you are coming at this from a different angle, but it would be great to hear specifically for your area what you feel the big blockers are that we need to remove to get to our vision of precision health. Maybe Jill, we'll start with you.
Yeah, sure. I mean, to me, the big blocker is evidence. It's 20 years since the completion of the Human Genome Project, and all of this promise of more personalized medicine and more precision medicine that was supposed to come of that initiative. We all go to these conferences all the time, right, where you list all of these reasons, all of these impediments to why genomics or precision medicine isn't being used more broadly. But I really think it boils down to evidence. And one of the challenges is just that the model that we have for these new startup companies, a lot of them are, investor-backed or venture-backed specifically. And they, come from, oftentimes the leadership comes from the tech industry. They're not familiar with the healthcare evidence standards that, in fact, some of them are actual regulations that you must have evidence for them. But even if it's not an overt regulation, if you don't have evidence that your test works or that the intervention based on that test is useful, drug companies, it's dictated by the FDA what the process is. And for traditional device companies, it's dictated by the FDA what the process is. There's just one and only one route to market, and that's evidence generation. In these laboratory-developed tests, which is a lot of what the precision medicine tests are, there are options for how you can go to market, and there's not real great oversight over making sure that the test works, analytical validity, or clinical validity. So I just think precision medicine just kind of needs to get in line with and generate the same kind of evidence that every other healthcare product has to generate. And then once they prove the utility, that is the biggest barrier. And so as a community, I just really feel like the most impactful thing we can do is focus on how we can help these test developers get the evidence that they need as quickly as possible.
Paldeep, do you agree? Do you align with that? Are there things you see a little differently? How do you think about the blockers?
Yeah, I certainly can see that point of view and agree with much, if not most of it. I see some things a little bit different from my perspective as a clinician, but most in line. I suppose the one thing I would say is when it comes to education, it's not simply about understanding the methodology of the test. The education comes in actually understanding why the result is useful and why it's meaningful. And actually, this is very hard for for a non-genetic specialist in medicine. They never had any teaching in genetics and never any education. For example, an obstetrician now there's universal recommendations of using non-invasive prenatal screening with cell-free DNA. They often struggle to understand what the implications of the results mean and what to do next, even though they understand the result is useful. Similar examples in many other aspects of medicine. The only other thing I would add is the issue of access. And access is a really big problem, and it comes back to it's intimately linked to the education part as well, I think. I mean, I see patients that they come and see me because their local genetics group, either they won't see them because they're an adult and they only see children, or they're put on a 3-year wait list. Now, I'm not actually joking, it's 3 years to be seen. and they tell me, I'm not waiting 3 years to be seen. So I'm going to travel across the country, or I'm going to make it happen. I see people internationally for the same reason, Canadians, Europeans, just because they don't want to wait years to be seen by a genetic expert. That's not something that any of us should be happy with, and I think it's okay for patients to wait. I don't have all the answers for that, of course, but I know there are some innovative solutions to be able to use it in a scalable fashion. And these are things like hereditary cancer screening, polygenic risk scores, pharmacogenomic testing, all of which there's plenty of evidence for, but the actual application and use of that is falling behind.
Yeah, no, I agree with your points about access to specialists. It can be challenging in our healthcare system, and genetics is certainly a specialty that can be difficult for people to get in and see them. So I agree with that. And we need better processes. And I think COVID actually is going to kind of help a lot with that in advancing more telehealth options. But now that I work on the payer side of the house, like it's really underscores to me the need for evidence. And I would actually say that polygenic risk scores do not yet there isn't a intervention that has been proven useful, around polygenic risk scores. I think there's some ongoing studies to try to see if they can get that to declare itself with certain, specific applications. And so for your example with OBGYNs and NIPT, if they really don't know what to do with those results and they're not using their, all those online positive predictive value calculators that they're supposed to be using, and the differences, some of those calculators have it even by the different manufacturers. If they're not using that and not using that properly, that's pretty dangerous. And I don't think we as a healthcare system should be encouraging that, right? So, if it's so complicated that the primary care doctor doesn't know what to do with the results, you really have to stop and ask yourself if that's the right setting to be implementing that kind of test. That it's probably better off done by a specialist if it's that complicated of a test. Again, just being controversial, just pushing the envelope here, making the conversation interesting.
Yeah, I think the argument with that, of course, is they are the specialist. They are the prenatal expert, and so back to my point, they're struggling with interpretation of the test, and I think we can agree to disagree on some of the evidence of FDA and pharmacogenomics, but you are right. There always has to be a maturation of that evidence and alignment, and we are seeing that as we get larger databases, larger data sets, and richer data.
So let me speak to that a little bit because I'm on the research side. So I am in that group that is trying to deliver stronger evidence for just support for genetic associations, because ultimately, based on those associations, that's how we design diagnostics. That's how therapeutics are often made. It is hard, and there's a lot of there's a little bit of the blame that we can put on researchers, because a lot of the research in genomics that has been done has just been like publishing associations. We've sequenced 100,000 people, 5,000 people with a disease, or just from like this specific ethnic group or from this hospital network. And here's like 50,000 associations that we have, or maybe even like one or a couple strong associations that we have, but that hasn't been validated. So there's been, for a long period of time, until very recently, it was very easy to just publish a paper, just publish like GWAS results, and that's it. And he's like, here's all those findings, but we're not going to take the next step and see what they mean and try to validate those findings in some type of experimental setup. But then it also comes from back to the difficulty of just like running those associations, because human genome is huge. It's extremely complicated. You need a certain type of power to make statistically significant findings, and that power will only come from clean databases. And that just doesn't exist, and we're nowhere near getting there, based at least on my experience. So we need data that is consistent in terms of like how it was collected, and that genetic data, phenotypic data. Ideally, we would love every study participants to be sequenced using exactly the same technology, the same library prep, and the same sequencing facility, because if it's done slightly differently or very differently, it introduces biases. We'd love them to be diagnosed by the same doctor over their lifespan, but that just doesn't exist. So the databases are just don't have consistent data, and it's very hard to compare between them. They're very incomplete. So just for researchers, until we clean this mess up a little bit, if I may say mess, we're not going to be able to produce stronger evidence.
As I think about the audience here, and even the three of you, we've gathered here the genomic community, and there's sort of almost a question in my mind of how do we all come together on these blockers, and particularly the data. It seems like, really being able to bring data sets together, having the data infrastructure, having the clean data, like that keeps coming up as a blocker. And it feels like something we as a genomics community almost need to come together to solve. Do you guys have ideas for how we could do that, or like first steps we should take?
Make them more diverse.
I'll look at,
for instance, what Paldeep said should resonate with everybody. Africa as a continent is more genetically diverse than the rest of the world combined. There's just like so much that we don't know and don't understand in genomics that we just have to make those data sets more diverse.
In reality, it costs money to build those data assets. And finding that pre-competitive space is hard. Trying to do it as like, a publicly paid effort, like a government paid program, is one way to kind of try to get around that. But those tend to move incredibly slowly. And so we're seeing people build a data asset, but we actually need to pool the data assets. And if anybody's got a solution for that, I think that's the big sticker. How do you solve the IP problem? And whose toys are more valuable than whose toys?
Yeah, and I think also if you look at internationally, we've seen some success. It's like the UK Biobank and others that they've made these things accessible, and that's actually helped tremendously with research as well. So there might be some sort of, the solution might actually be a hybrid of this private-public partnership, and that there is some aligned incentives. Ultimately, it's not a question of which data is more valuable. It's the answer is the data is more valuable together, and so having to figure out how to get everyone's incentivized and aligned to see that.
I agree. It's not going to be simple, and it probably will never happen. You're right. A lot of different players would have to agree on using some consistent rules of data collection and data sharing, which just is not going to happen. Plus, then you have a lot of groups, especially, somewhat vulnerable groups that are just very wary of sharing their data and making their data available for research, making their data available for kind of like anybody doing whatever they want with that. So a lot of those groups are just like not participating in large programs like the All of Us program, for instance. And we have to also address that. Like, we have to find ways to engage participants who are a little bit less trusting towards researchers or pharma because their data has been abused in the past, and find ways to give participant some control of the data. Which, there's a number of different data governance structures that have been proposed, and some of them work better than others. But it is possible. And when it comes to IP, and genetic data, I actually don't see an issue here at all. I don't think genetic data should ever be patentable. And I don't think we actually can, patent it in the States. We can derive patentable property way down the line when it comes to, developing a diagnostic test or developing a therapeutic intervention, but not in the data itself. So it's kind of just like, it's going to be much more down the line.
Is there one thing you would say to, the genomics community of like where we should rally? What would you say, Jill? Where should we rally to make this precision medicine thing become a reality?
Accept that you have to generate the evidence. Don't convince yourself that your pet test is so special that you don't need evidence. You need evidence. And then let's figure out how to partner to get you that evidence as quickly as possible.
Kaja, you kind of said that one of the places you'd love to see us rally is around diversity in our databases. What would be your hope? Like, if we do that, if we were able to do that today and get incredible diversity, 5 years from now, what would be your hope for what we can do with precision health?
I mean, then I think my hope would be we could address diseases in a way that's like more tailored to different ethnicities, because there's still a lot of issues with misdiagnosis or underdiagnosis where, if you're not of white European ancestry, it's pretty likely that the genetic variant you're being tested for comes back as a variant of unknown evidence. So this is going to be really, really hard for doctors to be able to like work with that data, interpret it, and deliver better health benefits to those communities. And for that, I think we do have to make sure that they are included in the research. But they're also the ones that are very often to be more hesitant to be included in the research. So as a community, we also have to find ways for study participants to benefit from the research more than they do. When we share our data with Google, we get access to Gmail. We share it with Facebook. We get to see, our high school's friends' kids' photos. But if we participate in clinical trials, we usually get access to free healthcare, at least for the duration of the trial. When we share our genetic data with genetic companies or with researchers, we don't really get anything out of that. So I think that also has to change. And those expectations towards people just like freely giving out their genetic data has to change.
Do you see anybody that's like created a model that you think could work on larger scale? Or do you think the models still aren't out there yet?
I think there's a number of models being tested, and I'm very excited to see what's going to come out of that. So we're testing one model. We're a benefit sharing company. We made a commitment to share 4% of our revenue with our study participant, with the participating communities, and they get to decide what those funds are going to be spent on. And we're very proud of this commitment. There's also companies like Luna DNA, where participants in research who share their genetic data get equity in the company. So there's a couple models out there, and I think we'll get to see which ones of them work and which ones resonate with participants.
Paldeep, what do you see as your great hope for precision healthcare? Looking 5 years out, what's your great hope?
I would like to see more democratization of access to evidence-based genomic testing, such that it's not dependent on the geographical location or the availability of a local genomics or genetic expert, and the right testing is always available based on the need. And it's not just reactive, but it's proactive in that there are safeguards or some monitoring in place to actively suggest particular tests or workflows, perhaps based on certain biomarkers or certain diagnostics or certain diagnoses that are coming up in someone's health. So just using the data, using the information a bit smarter and a bit more proactively.
I just want to say thank you to everybody. I really enjoyed hearing what you had to say today, and I appreciate you participating in the panel. Thank you.